This is episode 76 of the Immunology Podcast, Innate-Like T Lymphocytes with Dr. Mitchell Kronenberg. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Raub. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today we have Dr. Mitchell Kronenberg from Jolla Institute on the podcast to talk about his research on innate T cells and mucosal immunology. We've also got our usual roundup of recent highlights in immunology news coming up. But first,
we can't wait to attend Immunology 2024 in Chicago. The meeting takes place from May 3rd to 7th. And we'd like to remind everyone to make sure you've registered and that you have booked your hotel. Learn more at immunology2024.aai.org.
Got to start making dinner reservations. I promise I'll do it. I'm going to get on it.
You've been saying this,
I know.
Dear listeners. How long has Jason been saying this? I lost track. If you don't make these reservations, we're going to eat dish pizza every single night.
Okay, well, that's motivation right there. All right, I'm on it.
What is, I don't know, I think we've mentioned it before, but today we want to officially announce that we are having a live show.
We are, and it's on May 4th. Which is Star Wars Day, because may the 4th be with you.
Yeah, may the 4th be with us, indeed. Dear listeners, please don't miss it. Come over. May 4th, that's the second day of the conference, from 12:30 p.m. to 2 p.m. So right lunchtime. And we have a very special topic. We're going to be talking about AI, artificial intelligence, in immunology research. I think it's a very exciting topic. We've been thinking a lot about it. We have special guests, Joe Vinny and Ryan Peckner from Seismic Therapeutics. They use AI for their research and for their products, and they're very special guests, and they're going to be discussing with us what is in the future of AI, what is AI doing now for immunology research, what can we expect to do as the technology keeps evolving. I am kind of fairly new to this AI thing, so I'm very excited to be there and hear out what is, as a tool, as a companion for our research, what can AI do for us? What do you think, Jason?
Oh, it's super fascinating. I actually worked on building neural nets back in undergrad for a semester for some very early research. Been seeing where it's kind of shifted from that type of build out to these large language models, and then what AI is able to do in science is particularly interesting. So I'm very excited for this. This is right up my associated with GI alley, but not quite GI.
Don't miss out. May 4th at 12:30 to 2 p.m. Look for us, join us, and just as make it special. We're so excited. I'm so excited. Live show. This is so fancy.
All right. Well, do you have any papers you can segue off of Star Wars, or should we just get into it?
I have something that the Emperor would have, sold the galaxy to get.
Okay.
The Fountain of Youth.
Okay. All right. We'll use that as a segue. All right. So what Fountain of Youth do you have, and how are you going to help me in my arthritis, Brenda?
Well, in this publication, I'm going to talk first. This publication is called Depleting Myeloid-Biased Hematopoietic Stem Cells Rejuvenates Aged Immunity. It was published in Nature, 27th of March. First authors are Jason Ross and Lara Myers from the lab of Kim Hassenkrug and Irving Weissman at the Stanford University and the NIH. And yeah, I think this is not the first paper about, reducing the effects of aging that I've discussed. So it's becoming, I think, more and more popular as a research topic. I think also we understand more things, and I see kind of a shift, kind of a theme here. Basically, in this paper, they start with the observation, and this is kind of already known, that as we age, the output from our bone marrow, from the hematopoietic stem cells in our bone marrow, shifts And as we age, we start from having a balance, what they call balanced output of lymphoid and myeloid cells towards a more myeloid skewed output from our bone marrow. And so in young age, this idea that we have a balanced lymphoid and myeloid production promotes lymphopoiesis and therefore adaptive immune responses, and also limits the amount of myeloid cells that we make, which can be pro-inflammatory. So starting from this assumption that there's this shift in age, and that this shift is kind of in part a cause of these dysregulation that we see of the immune response with age. So as we age, we have less lymphocytes, we have more inflammatory markers, we have more myeloid cells, and these are all markers associated with that aged immune system, which also is an immune system that has a reduced capacity to respond, particularly to new pathogens in a timely manner, to mount effective and durable adaptive responses. And this is something that we observe in mice and also in humans. So what they propose is that by skewing back to a balanced output, this bone marrow that the composition of the bone marrow, we can potentially improve a little bit the balance in the lymphoid versus myeloid cells in aged organisms. So they start with mice, and they according to previous research, there is one marker, CD150, better known as SLAMF1, which is expressed on hematopoietic stem cells. And two populations can be within mouse HACs. Two different populations, CD150 high and CD150 low, can be identified. And this seems to differentiate cells that are this balanced, with this balanced phenotype, or this balanced output versus myeloid skewed. So the idea is that they want to identify markers that they could use to deplete myeloid skewed hematopoietic stem cells, and therefore generate a new bone marrow niche that has a more balanced lymphoid versus myeloid output. So they also start looking for other markers. I'm not sure if like CD150 is patented or something, but they also try to find other markers that associate with CD150, that associate with aged bone marrow, that they could potentially target and deplete. And they actually come up with other markers that I'm not super familiar with. They're called NEO1, CD41, CD62P. And these are all seem to be enriched in cells that are compatible with this myeloid, preferentially myeloid hematopoietic stem cells. And they show that they can deplete, using these markers, that they can deplete this population, and they can generate mice with kind of more balanced bone marrow composition and output. Interestingly, they tried CD150. They also tried this NEO1, CD62P, and they end up kind of using NEO1, which they actually deplete using goat antiserum. Again, I think this is something about some kind of IP. I don't know. It's not clear to me why they move away from CD150, which seems to not be a established marker, to these other NEO1 that they pick up from their own analyses. But they zero down on that one. And they show that by depleting this myeloid HSCs from the mice, they can kind of balance the output, increase the amount of lymphocytes versus myeloid cells in the mice. And when they actually test that at the end of the paper, they do a vaccination, a flu, I think it's flu vaccination, and they also seem to improve substantially the response of the mice after to the secondary, so the infection after vaccination. And they show that indeed, by depleting this excess myeloid progenitor, so to say, and helping the mice balance out their lymphoid versus myeloid compartments. This not only reduces inflammation. They look at IL-1 beta and other inflammation markers in the serum. They see a reduction of these, and they see some of the hallmarks of a younger immune system when it has to do with the amount of activated cells. They see an increase in naive phenotype cells, and then when they do an actual challenge, these mice after treatment respond better after vaccination and survive the actual infection substantially better than their not treated companions. So, yeah, maybe no. They also indicate that these markers, they're similar markers, are homologous markers in humans. So they are kind of suggest that this depletion could also work in humans and potentially also help aged human immune systems recover a bit in years and be more responsive and less inflammatory. So yeah, I don't know. It's feels too good to be true, kind of easy. I'm very curious to see how this kind of approaches fare in the long term. But yeah, you might just need to get rid of those pesky myeloid hematopoietic stem cells.
They're the only ones I have left.
That's also what I think, right? At some point, if you don't have a thymus, it's not like you're gonna make many more T cells, are you?
Well, I
So,
also apoptosed most of mine with 6-MP, so
well, that's another thing.
Yeah. Yeah. I don't know. That's interesting, but I agree with you. What's the clinical relevance will be the tough one here.
Yeah. Yeah, and also the mice that we're using, I don't think they were that old. So whether they actually like what kind of age this would kind of translate in human years, I don't think it's 60. I think it's like 40. So
Perfect.
well, you're right there.
All right. Well, moving
but-
on here. Half the time for a while we were talking COVID. And then I always talk microbiome sometimes. But there's another thing I get really excited to talk about when it comes up sometimes. What could that be?
I know the answer, but I'm not going to say it.
It's Gasdermin.
Yes, I know.
Our favorite pore former. We're back, folks, with another Gasdermin D paper. And it's interesting following the growing information on Gasdermin D that's been coming out, because when I was a postdoc, it was known but not super well described. Some of it, like its basics were there, and I was seeing weird stuff, and then seeing some of this weird stuff be manifested with understanding years later. It's exciting.
Mm-hmm.
Exciting to see the progress of science. All right, so in science immunology, we have the palmitylation of Gasdermin D directs its membrane translocation and pore formation during pyroposis. Only one first author. I apologize in advance. Aram Ugam Balasubramanian. Last author is Hongbo Lo. All right, so Gasdermin, right? It gets cleaved by caspases. It goes to the membrane. It makes pores. Pores open up. Cytokines go out, including the IL-1 pathway, really important for IL-1 beta release. And then other cytokines get released. It's very controlled, though. It doesn't just flood. And then sometimes the pores then let in a bunch of ions and it kills the cell. And sometimes it doesn't. And that's also controlled by other molecules. Cool. So it's like controlled pore formation and shenanigans ensue, but the shenanigans are also controlled. So what's neat here is they're like, well, we know about that, but we don't know necessarily what targets it to the membrane, what else is going on here. And one of the things that makes cells go to the membrane is by putting a big old fatty lipid on it. So palmitylation is the main way you do that one of the main ways you do that in mammalian cells. So they start doing some pull down assays, and this is mostly in vitro work or cell work. There's a few mouse studies with mice that I didn't have at the time. Very sad I didn't have these mice, but they now exist. So Gasdermin D, they first find with pull down assays, standard co-IPs and mass spec interacts with fatty acid synthetase and undergoes palmitylation. Cool. All right. So that's step one to get there. They identify, they then do some not inhibitors of palmitylation, so they can get a FASN, that fatty acid synthetase inhibitor. There's these chemical inhibitors. And they can show that throwing it into a macrophage systems, pyroposis is blocked. Okay, so polymerization is important for forming the pores and doing that. And they can see not only does the end state not work, they can see that pore formation is also, because you can make these fluorescent. You can kill the pore formation and that goes away. All right. Well, that's pretty cool. So that's basically the start of the paper. Then they identify that it's on cysteine 192 that's polymethylated. So they do some mutagenesis and look at that. Okay. And then so now we know where it's being polymethylated and that it's important for its function. So we have all that. And then it only translocates the cleaved form, not the full length, to the membrane. So they show that. And then they go, well, what polymethylates it? Because FSAD is not the polymethylator. It synthesizes the fatty acid, but you also got to slap it on the protein, right? It's just that they know these complexes exist where you have any protein, FASD, and then the polymorylation acetyltransferase enzymes, right? So you have to have the enzyme that then hooks it on, and that makes it a ternary complex. So these are ZDHHC proteins. Mouthful, right? But these zinc finger proteins that do that. And they identify, they look at different cell lines and which ones are low expressed in human cells, and they do some knockdowns of the ones that aren't at super low levels and multiple cell lines to find the same pattern over and over again, which is ZDHHC5 and 9 are the two that are important for gasdermin. And they verify that with some siRNA knockdown and kind of go through that whole thing. And then they're like, well, how is this all being regulated? And they actually find that LPS ahead of the full-blown inflammatory signal in inflammasome is critical for this. And there's some LPS NF-kappa B crosstalk because NF-kappa B inhibition inhibits this. But you have to have an LPS NF-kappa B signal ahead of a full-blown inflammasome signal to get this polymorylation to occur. It upregulates the enzymes that do this. It's sensitive to reaction to oxidative species as well, will help initiate this. But it's a precursor to the inflammasome going full out. And then the inflammasome goes full out, caspase activation, cleave and go. Right? So uncleaved will get polymethylated. And the cleavage is the last step, but it's not like it's cleaved, then polymethylated and goes up. You're priming the system by putting that tail on and then shipping it off. And then they show the variety of inflammasome signals that induce this, and you always got to have polymorylation for it to work. If you knock it out, it doesn't work. And then they do this in murine models of sepsis and show that, hey, gasdermin knockout reduces sepsis, or if you inhibit polymorylation, you can reduce sepsis both in a cecal puncture model and just an LPS model. So you can roll back time, you can prevent some sepsis death, pretty significant amounts, 100% to 50% type death. So half saved by blocking this cascade, by blocking with a specific, and they do a general inhibitor polymorylation, but then, and that has some effect, that gets you 70% death, but you can get down to 50 if you use a specific inhibitor. And so that was pretty cool to see that a very specific inhibitor could be even better. Sorry, 50 survived up to 70%. I got that flip. 50 survived baseline with pan inhibitor, a very specific molecule just for these that binds the gasdermin polymorylation complex and blocks it, gets you to 70% survival. So then there's a survival benefit, and you're starting to think therapeutic. Someone comes in septic, so you give it to them once, maybe, slow down the inflammasome process, the pyroposis, and maybe save people. So there you go.
I'm so happy that you got to talk about gasdermin D again, Jason. It makes me happy to see you so enthusiastic.
And it's not about epithelial cells.
That's true, at least in some proper macrophages, But it's funny because there's so much about the lipid metabolism in macrophages that it's so important for their function. I'm always surprised that we find a different, one more angle why, lipid syntheses or things like that are super crucial for macrophage in general. And I think it's very interesting that you can actually find specific acetyltransferases that will block, because I can imagine that many other proteins are being palmitoylated in a macrophage. But this ones that they find that there's this inhibitors that they use seem to be more or less specific for this particular cysteine in gasdermin.
No, it's super interesting they were able to find this one.
Yeah, it's like quite a found, because I can imagine that many other proteins also get palmitoylated, and then if you start just you don't have a specific inhibitors, it would be quite really hard to pull apart the mechanism. But well, very good, very interesting as always. Um, so for my second contribution to today's roundup, I have a smaller story, but I thought it was very nice. Also because sometimes um I think this is an example of sometimes a simpler approach can be better, even if it means not using fancy genetically engineered T cells to solve your cancer. Sometimes I have to accept that just a good, you know, antibody conjugated compound can do the job just as well and even better. And this is an example where sometimes what you're targeting makes can be very important. So this paper is called TRBC1 targeting antibody drug conjugates for the treatment of T cell cancers. First author Tushar Ni- Nishikawada. I'm sorry, Tushar. I hope by this was not too bad. From corresponding author Suman Paul from the Johns Hopkins University. And so for T cell cancer, uh, cancers that are derived from T cells, um, are a little bit complicated to treat. So B cell leukemias, for example, is nice. We have CAR T cells. We can target CD19. So in those really desperate cases, you can always just get rid of every CD19 positive cells. And even though you don't have any more B cells, you can get around that. People can be treated with immunoglobulin infusion, so people can still survive this, and at least they don't have cancer. So well, in the case of T cells, you cannot really do that. So you cannot target kind of pan T cell antigens because you will get immunosuppression. Um, but there is one interesting characteristic of the T cell receptor, which is that for the beta chain, there are actually two isoforms. TRBC1 and TRBC2 are the two types of genes that code for the constant region of the TCR beta chain. Interestingly, there is a tiny difference there's I think it's like 4 amino acids or 6 amino acids difference, and this actually generates an epitope that is against which we have antibodies for the case of the isoform 1. So TRBC1 isoform has actually a little epitope that can be targeted with antibodies. So there is some interest in thinking, well, if you are lucky enough that your cancer has a TRBC1 isoform, we could potentially get rid of every TRBC1 T cell, and you're probably still going to be fine. So there's actually has been a phase 1 clinical trial testing the safety and efficacy of an anti-TRBC1 CAR T cell. However, uh, the problem with this, what it was realized is that uh, in most cases, patients infused with the cells actually, um, most cases, the CAR T cells disappeared, and they could not be detected in the peripheral blood of most patients. And this is kind of critical. You need the T cells you need the CAR T cells to expand and to grow in numbers and have some level of permanence in order to get rid of every cancer cell. And so what turns out is that what probably is happening is that although the leukemia cells are probably not what's the word cytotoxic, when the CAR T cells also target or that about half of your T cells that do have this TRBC1 isoform, you're kind of activating the TCR of a T cell. And you're kind of making a synapse with a TCR of a T cell, which most of the time will result in that T cell killing you. So probably that's what's happening is that you are, by having a two-way activation, yes, the CAR T cells are probably attacking whatever they're targeting, but many times that cell is attacking back, especially if it's some kind of memory cell, some kind of previously activated T cell. And this is how this is probably preventing your CAR T cells from, really proliferating and taking hold. So, well, this shows that sometimes the fancy CAR T cells are not necessarily the answer, and how forgetting that we actually have a fully, normal T cell compartment that can attack the cells, forgetting that cost the result of this clinical trial. So what they say is that, well, it's still good to target TRBC1, but let's take a step back and then use a different approach. So they actually do an antibody drug conjugate, and they show that by targeting TRBC1, they can actually internalize, so they can generate an internalization of the antibody. They do that with some fluorescence microscopy. They show that eventually the TRBC1 antibody colocalizes with LAMP1 in lysosomes. So they can really kind of induce uptake, because we know also that TCRs are internalized upon activation. So you can really take advantage of that. And so basically they test a couple of drug conjugates that they already knew could be good targets, could be good candidates. And they show that one particular drug called SG3199 can be potently kill these leukemia cells when it's conjugated to a TRBC1 specific antibody. And they do some in vitro studies, and they show that they can, in vitro and in vivo, they can also, these antibodies can kill both regular TRBC1 cells, but most importantly, the cancer cells. And so that can actually be an interesting approach to targeting a TRBC1 positive T cell cancer. And they do some xenografts, and they show some level of success. So I think that's very interesting. So they seem that they have also generated a human IgG1 antibody that is chimeric with this anti-TRBC1. So I think they are, really planning on testing this in human subjects.
Yeah, I mean, you don't dismiss the antibody drugs conjugates. The ADCs are like current gen medicine that's starting to really come into its own.
Yep.
That's cool. Yeah, because all the T cells being activated is probably not good for your health.
Yeah. It's kind of funny. So I was like, when I was reading, it's like, yeah, of course that happened. It's like, oh,
Wah, wah, wah.
pretty much.
All right.
Okay, what's up then?
Well, big phase shift. Let's talk about activating everything in one final go with this virus. We're going to talk about Crimean Congo hemorrhagic fever.
Sounds scary.
Virus uses LDLR to bind and enter host cells. Nature Microbiology. First author is Vanessa Montiel. Last author is Ali Mirazimi. All right. So this is one of those emerging infections that make people not feel so good about some things. It is the Crimean Congo hemorrhagic fever virus. It is transmitted by ticks, and as the temperature gets warmer, it's coming into Europe potentially. Yay! Hemorrhagic fever virus. Those who don't know hemorrhagic fever virus, it goes to your endothelial cells, kills them, and then you bleed everywhere and die. So that being said, I don't have to spend a lot of time talking about this paper because it pretty much cuts the chase. For one of the things we like to know in virology is how a virus gets into a cell, right? So we know herpes zoster, VZV, or VSV as well. I think. Let me make sure I get this right, because they use that as the VSV. Sorry, is herpes simplex uses the LDL receptor. Well, they did a haploid screen and found out that this virus uses the LDL receptor to get in. Okay. So they do that. Then they really show it, right? So they show it by making fusion viruses, which are the VSV with the protein they think is important for this on the outside, just to be like, well, if I put this protein on a different virus, does it go in? The answer is yes. We could discuss the ethics of it. I don't think they were creating Franken viruses here. They were using, mostly dead things and putting a protein on to let it get into a cell that the virus could already get into a different way, right? So they really they weren't giving it any new function. They were just playing hot swap to show that those glycoproteins are important. So they identified the glycoproteins involved. They showed that LDR knockdown cells had decrease in infection. They show that it binds this protein GC, that's glycoprotein, binds and induces endocytosis. They use this using a bioluminescence resonance energy transfer, so a fluorophore into a luciferase, Brett, the brother of Fret. So they use Bodipi labeled LDL and then a NLUC LDLR receptor. Long and short, they show it induces endocytosis. They take it all the way through. They go into exhaustive detail, really showing this. They show that soluble LDL receptor blocks infections in cell culture. They do blood vessel organoids and do that. They realize the regular organoid structure has the wrong part on the inside because organoids are, grown in matrigel inside out. So they flatten it out and do monoculture layers on matrigel afterwards to make a 2D system so you can get to the right side. So they solve that and show that too, which is like a little neat thing that we know in other systems that they show here. And LDL knockout delays the disease in mice. And then they go into human isolates and show that it's also the important receptor in several human isolates, like from a Turkish patient. And they also show that ApoE antibody blocks the effect of VSV, but not this. So it's not important for ApoE. ApoE is not important here, unlike with VSV. And they also show that LPR8, which is an LDLR-like receptor, is also a receptor for this. So some receptors in the same family that are closely related by homology also work. So if you are in the virology field or interested in LDL receptors in virology, this is a great paper, kind of gives you a sense of what to go after. Also means it's druggable, which is good. In case, we all get this from ticks in Europe in a few years.
Oh my God.
But you're too far north for ticks.
Yeah, I think so. But no, but they're getting closer, huh? Yeah, I know the ticks are coming. I had to say, the more I hear about ticks, the more I just like
we have all over the place where I am.
Yeah, I know.
Tick Central in Pennsylvania.
Oh my God.
Yeah.
All right. Well, let's hope we don't have to use this knowledge.
But if we do need them, we always have our Innate-Like lymphocytes coming on board. And we're going to be talking to Dr. Mitchell Kronenberg at the Jolla Institute in just a moment about just those types of cells. But you know what else? Are you interested in improving your T-cell therapy development? With STEMCELL Technologies' new GMP manufactured Immunocolt XF, you can achieve robust T-cell expansion with high cell yield and viability without the use of serum. To learn more, visit www.stemcell.com/immunocolt-XF for that GMP manufacturing, which is awesome.
For the second part of our show, for our interview, we have, as always, a very special guest, Dr. Mitchell Kronenberg. Dr. Mitchell Kronenberg is a professor and President Emeritus at La Jolla Institute for Immunology. He was president for many years, so great experience. And La Jolla is one of those very iconic places to do immunology, especially in the US. And we have very excited to hear about his experience there, but also about their research in his lab. Dr. Kronenberg's lab works on unconventional T cells, for example, MAIT T cells. We actually did cover a paper of his lab recently, so we're hoping to talk a little bit about the different subsets of MAIT T cells in humans and in mice. Dr. Kronenberg, thank you so much for joining us today. We're very excited to have you on.
Oh, thank you. I'm happy to be here.
Thank you for being here. All right, Brenda, it's T cells. Do you want to go first? Do you want me to jump in because it's weird T cells?
You know what? Go for it. I see that you're eager.
I am. All right. So one of the things I've said on the podcast for a while now is that T cells are like, we always have these different names for, like, you have innate lymphocytes and you have MAIT cells and you have ILCs and all of that. And I'm like, how much of it are they different, or is there a skid mark of continuity? So, and they're all kind of related to each other in different steps on a ladder. So I was wondering if you could start, since we're talking about these unconventional T cells, if you could describe maybe the main flavors of ice cream, or the main rungs in the ladder, and how they relate to each other in terms of, these different types, like a MAIT cells, like a GD-T cell in this way or not, if you had them on a spectrum, or how you'd kind of nest them in relation to each other with a little overview of the flavors. Because I think it's a landscape that keeps growing quite a bit.
Yeah, thanks, Jason. I agree with you. I think there is a gradient of innateness. And actually, there's a paper on this from 2019 from a group in Boston led by, I believe, Patrick Brennan, where they looked at human cell types, gamma deltas, MAITs, NKT cells, and compared them to NK cells, which are true innate lymphocytes. They don't have RAG proteins, and CD4 and CD8 T cells. And they even found that memory cells have some innateness qualities on this gradient that they made. It's called a mixed linear model. And we've now done a similar analysis in mice. It's been submitted. It's not accepted yet, and have found the same thing. So these innate T cells, they are different certainly from naive T cells. They have things in common. They seem to be less proliferative, tissue-seeking. They can respond to cytokines without using their antigen receptor. And some of these properties you see to some extent in T-resident memory cells, even from the mainstream of CD4 and CD8. So a gradient is a good way to look at these cells. And I do believe that they do have a number of things in common. And then if you drill down further, I believe that the NKT cells and the MAIT cells, which are alpha beta T cells, have a little more in common with each other than compared to the gamma delta cells, which are a little further out. So for example, the MAIT and the NKT cells are positively selected by double positive thymocytes. They need SLAM proteins and SLAM interactions in order to develop, and so on, things that are less evident in the gamma deltas.
So in the case of MAIT cells, so one of the things that it's, when we think about T cells, alpha beta T cells, is this idea that they can only recognize things expressed by basically your own immune system. They're designed to respond to antigens being presented by your very polymorphic MHC proteins. But then in the case of MAIT cells, they are actually responding to fairly conserved MR1 MHC like protein. But they do have like just particular rearrangements of their V alpha chain. So it does look like a proper TCR, just that didn't go out through, doesn't have the level of polymorphism as a regular TCR. And also many times in this case, now I'm coming to your paper on MAIT1 and MAIT17 cells. There are a lot of overlaps in the kind of cytokines they produce, the kind of niches they occupy with proper alpha beta T cells. And I think it's always the question is, why this redundancy? What do you think is the evolutionary purpose of having these two types of similar but not really cells that they overlap in so many things? Why?
Yeah. That in some ways is a profound question.
No pressure.
In a way, these cells are, I look at them almost as a branch of innate immunity. Some people say they bridge innate and adaptive immunity, and that may be okay, because they're usually not the first responders. Let's put it in that context. In other words, epithelial cells or other kinds of myeloid cells that make things like IL-25, IL-33, TSLP, IL-12, IL-18, though, or that have, or IL-Cs that have intense neuronal connections. Well, IL-Cs really aren't first responders either. But so the non-lymphoid compartment are generally the very first responders. But I look at these cells as being innate-like in some ways because they respond very quickly and with effector cytokines, and they do a kind of what you might almost call pattern recognition. If you think of TLR4, it sees LPS. It doesn't see one thing because there's different kinds of LPS, but basically it sees LPS. So MAIT cells see riboflavin metabolites that are microbial specific, that are in bacteria, many bacteria, not all bacteria, and that are in some fungi as well. NKT cells see particular kinds of usually diacylated glycolipids, in other words, 2 fatty acids and some kind of sugar attachment. That again is, there's more overlap in the NKT cell case between the microbe and self. In other words, they're more overtly self-reactive. But they're still basically microbial products because of the way the sugar is connected to the fat. Okay, the stereochemistry of the linkage is more microbial, and is in fact found in Bacteroides and other elements of the commensal microbiome. So in other words, these highly conserved T cell receptors, which do have to rearrange randomly, if you will, and do have to undergo selection, they see microbial patterns using non-polymorphic MHC molecules. And so the analogy to innate recognition or pattern recognition is very, I think it's very clear. And so why have that? Well, the immune system is redundant, right? Has layers and layers. And it's almost as if the adaptive branch, the T cell branch, decided, hey, innate immunity is really good. We want to play in that field as well. And I'm anthropomorphizing it, if you will. But it's almost like that. Why not have a T cell, a branch of T cells that do things similar to innate immunities in case that there aren't enough neutrophils or macrophages around, or and we need a good shot of gamma interferon early on, and nobody else is going to do that.
So do you think then we know the classic innate and adaptive crossplay, like innate sees it, right? Really epithelial cells see it. Let's give it to the hands out to the original innate cells, which is my joke to Brenda. But then the innate immunity sees it, and then the adaptive takes over. But the adaptive is also going to go back and like quarterback for the innate a little bit and help them really get going in the right way. So for these alternative lymphocytes, then do you think they're like the first quarterback, right? They're the quarterbacks that you're going to use to really potentiate the innate system until the adaptive system comes online, which we know is slower and later. Do you think that's part of their niche function? Is what we think of T cells doing for macrophages, but instead of having to be receptor specific and all that, it's pattern specific. So it gets that ball rolling without the delay.
I think that's well put. That when these cells get activated, they can have an adjuvant-like function on, for example, dendritic cells, and that will then drive the CD8 response or the adaptive response, as well as other responses. Ralph Steinman worked in this area a bit before he died, the Nobel laureate for dendritic cells, of course, and he felt that alpha-galactosyl ceramide, a glycolipid that activates NKT cells, was the best, Th1 CD8 adjuvant he had ever tested. He told me that personally, and other people as well have used that. It's a very potent glycolipid for NKT cells. Works in mouse, works in human, and it does really activate cross-presentation and CD8 responses very effectively. And there's also a paper from Paul Kleinerman's group in Oxford showing that MAIT cells are very effective adjuvants by communicating with macrophages and other cell types using an Oxford-generated adenovirus vaccine platform that they use. So yes, it's not just simply okay. We gave some, we made a response fairly early, responding to the very first responders, and we saw a microbial pattern. But it's also a matter of talking back to the dendritic cells and stimulating adaptive immunity.
All right. So I'm going to double up here. So mucosal immunology time then. So I think about a lot of these cells hanging out in the mucosa, which is my favorite place to be. I know you've worked in the IBD space and others. Could you give us a sense, maybe immune system is important, right? But where things start going wrong and what we're starting to understand about their central role in disease at that mucosal surface, whether it's IBD or otherwise, what these cells we're really understanding these cells are up to.
Yeah, they're certainly in a lot of tissues, and but their roles are a little bit complex, unfortunately, and therefore, when you look in the literature, you'll often see contradictory descriptions of their role in type 1 diabetes or their role in IBD or so on and so forth. And so it's very hard to give a simple answer. One of the things that's emerged, for example, about human MAIT cells, and actually to some extent mouse MAIT cells, I think I need to include both here. Yeah, they make Th1 cytokines. They can make gamma interferon. That's very typical especially for human MAIT cells. They can make IL-17. In human, generally, it's fewer than making gamma interferon. And unlike in the mouse, they tend to have a mixed Th1, Th17 signature. However, they also can exhibit a tissue healing gene expression program, amphoregulin, and other things of that sort. And there are actually experiments in the mouse carried out by the Belkate group in skin and in human, indicating tissue healing function. So sometimes you'll see a transcriptome and you'll go, oh, okay, I don't know if all those genes in the gene set analysis, how they relate to tissue healing, but there's actually experiments indicating that. And therefore, it's a little hard to give you a simple answer. So, for example, MAIT cells in human inflammatory bowel disease are activated, but they've also been reported to make IL-22, a cytokine that can have an epithelial regenerative capacity. So we're really not certain. In Crohn's disease, again, increased, activated, but not necessarily promoting disease. I'm talking about human samples now. In ulcerative colitis, in work that we discussed briefly before we started talking, we saw very few MAIT. And I'm thinking that maybe those two diseases, well, we know they're different. So in one case, maybe the MAITs are doing something in Crohn's, and here I'm speculating. And in ulcerative colitis, maybe they're much less important. I just went out on a limb, so don't saw it off, so I fall on the ground. But that may be the case. So no simple answer, but generally, if you have to make a broad generalization, pro-inflammatory and generally not good for autoimmune disease. Definitely activated during acute respiratory syndrome and SARS-CoV infections. Definitely not a good player there. Decreased in blood, increased in lung, making inflammatory cytokines. That's not good for seriously ill people with COVID-19. So a broad generalization that when you dig into the weeds doesn't fully apply in terms of the vector, if you will, the direction that they act in.
I like that observation that they can also seem to uh, help to tissue regeneration or all these other, because oftentimes we only think about, aggressive T cells or So I think it's so important to also keep that in mind, and that those other angles that these cells can have. I think it's so important, and this is not, of course, not the first time I hear it, but I think it's becoming more and more clear as new research comes up.
I think this is true for all these innate-like T cells and for ILCs, which are not T cells,
Yeah.
that they have roles in tissue homeostasis. ILC2, of course, amphiregulin and other things related to tissue healing. Gamma delta cells were reported many years ago by Wendy Hafren to make keratinocyte growth factor and participate in gut and skin healing. Beautiful work that she did. And so I think this is a theme for all these cells. They're tissue resident, and the tissue healing part of what they do is very important.
So, if I may change topics a little bit, what's also very important is to have the spaces to do this kind of research. And we have already actually interviewed some of your some of your colleagues at the Jolla Institute. We have Alessandro Sette also with us in the podcast. Um, but you have you have been at La Jolla for many years, and you were actually president of the institute for you mentioned 18 years. Um,
Mhm.
so maybe especially for those that are not so familiar with the La Jolla as a research institute, it is very iconic. But I think especially for those maybe that are from even farther away, like myself, um, La Jolla is an institute, kind of a self-standing institution, but has had so many what's the word, important contributions to our understanding of immunology. It has been such a great place to do original immunology research. So, would you like to share with our listeners, for those who don't know, what this institute is, a little bit about the story, and how it is for you to be part of this kind of place?
Yeah, thank you. Uh, well, to put it in the slightly bigger context, we have this region of San Diego called the Torrey Pines Mesa. And the Torrey Pines Mesa is totally devoted to research. And the UC San Diego is here, they're the behemoth. But there's also the Salk Institute, the Sanford Burnham Prebys Institute, of course, the Scripps Research Institute, which is the oldest of the independent research institutes, and in fact, the biggest. And then other organizations, the Sanford Consortium for Regenerative Medicine, which is a cooperative effort. So this is a very intensely talented area for all these different institutions. La Jolla Institute for Immunology we have about 25 laboratories, about 400 something employees, and we're about 35 years old. And the thing that attracted me here, I was a tenured professor at the University of California in Los Angeles. The thing that attracted me here was the focus of the mission and the lack of bureaucracy. And I think independent research institutes can be very special in that way, not just us, but and our other institutes here on the Mesa and institutes throughout the world. They can be really special places. And I had all my training prior to UCLA at Caltech. And Caltech, California Institute of Technology, it only has about 2,000 students total. It's really a very effective research institute, kind of masquerading as an educational institute. And so for me, coming from UCLA, which was a wonderful time and a great university, there's no question about it, but the load of bureaucracy and the different missions that UCLA has to do in educating 40,000 students and so on, the coming here to La Jolla Institute was a little bit like going back to Caltech, to having that pure focus on let's do great science, and everybody, whether you're changing mouse cages or you're doing maintaining the building, that's the mission. Everybody internalizes that and knows that. So I think that's part of the secret of this place. We have two partners that have helped to support us besides grants. We do write grants and depend on them as much as just about anyone else. But we've always had we've always had one partner, which is a Japanese company called Kyowa Kirin. They're a mid-sized company. They actually do research in the United States. so that they're in our building. There's 40-something employees. The holding company of this company makes beer. They make Kirin beer. So if you go to a Japanese restaurant and you order beer, you can have Sapporo. Order Kirin. You're helping immunology research. Okay. So, but seriously, they helped to start this institute with funding with a man named Kimi Ishizaka, who discovered IgE. And their support has been the equivalent of an endowment. They've given us the money to do things that we couldn't otherwise do, such as supporting a good part of faculty salaries and things of that kind. And in return, they get the right to license some of our IP. It's a very unique academic industry partnership. It's over 30 years old. They actually built our building with their 40-something people who work here. We manage the building. The building stands on UCSD land. So our other key partner is the University of California, and we teach there, and we have students, we share core facilities and so on. A place of this size, isolated in a beautiful place in Big Sur or whatever, which would be nice, right? Except for the road collapsing, but would be too isolated. So here we have access not only to our own colleagues, but our UCSD colleagues and UCSD core facilities. We can't have every single core facility. We're not big enough. We're great at imaging and sequencing and so on, but we can't do everything. We don't have EM. We only recently started mass spec. We're getting very good at it, but we weren't a few years ago, so on and so forth. So our partners are so critical for our own success. And yeah, that's been our mantra. Just do outstanding science and we'll get out of your way. And that was, I didn't come here to be the president of the institute. I came from UCLA, get out of the traffic, breathe fresh air and have great colleagues. But when my predecessor, Howard Gray, retired, there were some people, he wanted me to be his successor and I agreed to do it. It was fun to run the place. I had enormous freedom. I had a board of directors that would occasionally beat me up a little bit, but they were mostly my friends. And I had enormous freedom. It wasn't like being a department chair. We could really make decisions. Of course, you have to have faculty consensus, but what a great experience. And then after 18 years, or after 15, 16 years, actually, it took a little while to make the transition. It was, I think I had enough. And I wanted to go back to the lab and spend more time with my students and postdocs and try to make some more translational discoveries, directing more to human immunology.
So you mentioned something really interesting for me, and this comes from more my business side of, being able to be very mission focused without a bunch of split missions and having that advantage. And yet also you're on USCD, USSD land, you have this corporate arrangement on the other side. So it sounds like part of the special sauce of La Jolla is the ability to navigate and create those healthy relationships without the further, I'd say, bureaucracy, dendrites getting in or something. Do you have any, I guess, sense of wisdom or anything to share that you can share? Because it sounds like very healthy in a very good way to have partnerships without being overloaded with bureaucracy that comes with them. Is there something that you think La Jolla has done particularly well to create that? Is it serendipity, which sometimes is how the world works? But it sounds like quite the victory in a positive way, that you can kind of have all of these different things and not be filling out paperwork all day long to move a mouse from the UC side over to the industry side and be stuck in bureaucracy.
Yeah, I think what you said was insightful, and serendipity certainly plays a role. Kirin, in the early days, they're about 9% of our budget, but in the early days, they were 80%. When Kimi Ishizaka, in the 35 years ago, moved from Johns Hopkins, the discoverer of IgE, an icon in Japan. I think Kimi's joining this embryonic institute hooked Kyowa Kirin. It was called Kirin Pharmaceutical back then. And the stereotype is Japanese companies take the long view, and they have, to their credit, because we have now given them a very good asset that's in phase 3 trial, and we're hopeful for a lot of royalties for them and maybe some for us as well. We're not there yet. It's for atopic dermatitis, a blocking antibody that they made. But the IP comes from Mick Croft in our institute. Yeah, serendipity is part of it. I think, look, in my own experience, I may be patting myself on the back here. Developing these relationships means listening to the partner and not being, well, I'm an academic, I know everything, I'll take a little bit of your money and just go away and don't bother me. Okay, So we find ways to try to be useful to Kyowa Kirin and to try to be useful for the university as well, to be a good partner. For example, working with Steve Hedrick, a good friend of mine, we went to BioLegend and to Jean Lay, and we said, we need money, fellowship money for immunology graduate students. And we got it because BioLegend and Jean Lay is very, very generous. The point is that, okay, I helped, and Jolla Institute helped raise money for UCSD graduate students to support fellowships. So you have to give back, and you have to listen. It's just like any other partnership. And I think we've done that, not exclusively me, of course, but and certainly Erica Sapphire and Shane Cridy, my successors. We've done that very well.
I guess being resourceful, like always finding ways to interact with people, and there's always a way of improving and talking and being receptive to other people is super important. I think that we can see that from you, and that's always a good learning for anybody listening.
Yeah, thank you.
So, wrapping out our conversation, we like to also ask our guests some questions to get them to know get to know them a little bit better, and for our audience to get to know them a little bit better. And one of the questions we often ask is, if you were not a scientist, what do you think you would be?
Yes. I'm not certain. I think what I would like to be is an actor, and particularly a comic actor. Uh, I can spontaneously at times make people laugh. I don't think I can tell a joke that's written down very well. So I never had the courage really to pursue this in a serious way. I like attention. I don't mind speaking. Some of my colleagues, I have to give a talk. They're nervous. That's a pleasure. What an honor that somebody wants to hear
To
about my
hear you.
science. And as president of the institute, I had to go to different places and talk to people who I may be very different from. I may not agree with them politically, whatever, and I have to explain what the heck immunology is pre-COVID. People weren't thinking about B cells and T cells before COVID, and explaining is good. So I've done things amateur, very amateur, very sophomoric things. At Caltech, we used to write plays for the students when they graduated, and I was one of the go-to people for writing these plays and starring in them. This is truly amateur at the lowest level. But and honestly, I lived in LA for many years at Caltech and UCLA, and the entertainment industry and its glamour were very, very evident. And it always seemed like that was a different kind of life. Now, of course, people who do that, most of them don't have health insurance. They're starving. They're waiters, and they live a difficult life. Of course, yeah, if you're Ryan Gosling or whomever, it's all it's all glitter. And I guess at the end of the day, I, given those two options, if you will, I think I needed a little more intellectual stimulation than I needed to strive for that kind of creativity and perhaps adulation. So that's where I ended up where I am. And I'm happy with that. But I sometimes think about that creative side and doing something else, even writing a play, something of that kind. Never done it.
Well, or you can learn to, if you play any instruments, you can join one of the scientists, music bands, play something with. We had Jim Allison on the podcast some time ago, and he plays. He really likes that
I
too.
know.
So,
Yeah. I don't think, I love music, but I don't think I have the ear. I don't think I'm musical enough. I think it would be something in another format. But yeah, the creative side you often see in scientists, and it's wonderful.
well, we should have maybe in the next AI conference, we should have an open mic evening.
Right. Yeah, I've got some material.
I have to bring my
Perfect.
guitar, Brenda.
Excellent. See, now we can have some kind of little play with you in the background, putting some music. Excellent. So we'll let you know. Our agent will contact yours. So we'll let you.
Yeah, contact my people. Right.
All right. Well, thank you very much for coming on. This is fascinating. Starting to dive into the realm of non-canonical T cells. It was a pleasure. If you have any openings, you can let people know if you're looking for postdocs or anything else, or how to get a hold of you.
Well, Mitch at lji.org. I'd love to hear from you. Currently not looking, but a lot of people in the Institute are. And by the way, for both of you, if you're in San Diego, let me know.
I absolutely will. I come out to Irvine not infrequently for work.
I've never been to the West Coast. I need to make it. I know.
And you've been many places. Yeah.
I know. You know what? I think what I'm going to do is I'm just going to start contacting everybody I know from the West Coast and just like go from starting from what is south, like Los Angeles, all the way to Seattle and see how many people I meet on the way. Such a pleasure talking to you, Mitchell. Thank you so much for joining us. And we're looking forward to your upcoming publications and whatever comes next from your side.
Okay.
So,
thank you both.
Thank
really
you.
enjoyed it.
So again, Mitchell Kronenberg from the Jolla Institute of Immunology.
That brings us to the end of our show. Don't forget to subscribe to our newsletter, www.immunologypodcast.com, to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at @Immunopodcast or via email info@immunologypodcast.com with feedback or suggest guests. See you next time.